Welcome to the PharmaSource podcast. We're starting today with breaking news that's rocking the pharma industry and could change America's pandemic preparedness for years to come. Yesterday, August the 5th, US Health Secretary Robert F. Kennedy Jr. dropped the bombshell that the Department of Health and Human Services is pulling the plug on $500 million in mRNA vaccine funding. 22 active projects are cancelled, essentially bringing one of the most promising vaccine technologies to a grinding halt in America. What's actually happening here? The Biomedical Advanced Research Development Authority, BARDA, is winding down contracts with some of the biggest names in pharma. Sanofi, Pfizer, Moderna's bird flu vaccine program will all go, along with others. They've even told BARDA's investment arm to stop all mRNA-based investments entirely. For those following the science, Kennedy's reasoning is pretty extraordinary. He's claiming that mRNA vaccines fail to protect against upper respiratory infections like COVID and flu, despite the fact that these same vaccines are widely credited with saving millions of lives through the pandemic. An HHS spokesperson went even further, saying, "Until we can prove that mRNA technology is safe and effective, we have to focus on other platforms." Let's think about that for a moment. We're talking about the technology that was absolutely central in our COVID response. These vaccines were tested on tens of thousands of people and deployed at unprecedented speed. What's really concerning here is what this means for future pandemic preparedness. The whole advantage of mRNA vaccines is speed. We can design and manufacture them in weeks, not years. That's exactly why they were so crucial during COVID. And it's worth noting that this isn't happening in isolation. Since Kennedy took office, he's been systematically reshaping America's vaccine landscape. He fired all 17 members of the CDC's Vaccine Advisory Panel and replaced them with 8 new appointees. According to analysis by Science magazine, 4 of these new members have never even published research on vaccines. He's also removed COVID vaccines from the CDC's recommended schedule and has allegedly ended dozens of NIH grants that were studying vaccine hesitancy. For our industry, the implications are massive. Companies that have poured resources into mRNA platforms are suddenly facing huge uncertainty about the US market, at a time when threats like bird flu are potentially gaining pandemic potential. So perhaps the big question facing the global pharma community is, how do we keep pushing vaccine science forward when the world's largest healthcare market is backing away from one of our most promising technologies? Up next, I'll be speaking to Ben Locwin, Vice President of Reliant Life Sciences, to explain why mRNA therapeutics are so important, not just to vaccines, but also to cancer, genetic disorders, and other therapeutic areas. It's worth noting this interview was recorded before yesterday's announcement, so the tone about the future was a bit more positive than it would have been this morning.
mRNA, or messenger RNA, as the listeners may know it as, it was originally discovered back in the 60s. So we're talking 6+ decades ago now. There was a Nobel Prize awarded in physiology or medicine for the discovery. And ever since then, so over those past 6 decades, scientists have been looking at mRNA for its potential to be used in a number of different ways. Harness it to prevent and treat a wide range of diseases. As listeners probably remember well, mRNA technology really ramped up in earnest, at least in the public consciousness during the COVID-19 pandemic, as the vaccination technology that would allow rapid development of antigen profiles very quickly. The idea behind it being that the mRNA molecule itself contains instructions, or the recipe, as it were, that directs cells to make proteins using their own inherent natural machinery. How that also works in the case of, let's say, the mRNA vaccines, you've got to get it into the cells. So to facilitate this cellular entry, lipid nanoparticles have been used as a protective delivery vehicle. But then the idea being, with what it is and what it does, once inside, our cells read the messenger RNA as a set of instructions, and then they build proteins that match up with parts of the pathogen called antigens. And the immune system sees these foreign antigens as invaders, and they mobilize antibodies like T cells and train the immune system for potential future infections. And this, it's been really enormously successful. Moderna, for example, had a billion dose push with their mRNA vaccine internally that they were trying to go for. But it's been, a long time coming, really. So though the public only heard about it in the past few years,
Hmm.
it's been a long time. Um, I remember the first successful transfection of an mRNA packaged with a liposomal nanoparticle was published back in 1989. And then I think the next year or two, they took naked, unprotected lab-made mRNA that they injected into the muscle of mice.
Hmm.
And those became kind of the groundwork, which showed that in vitro transcribed mRNA with a chosen gene could deliver the genetic information to produce a desired protein within living cells. And that's what really led to this idea of, we've got a concept now for messenger RNA vaccines. And, they certainly have proven themselves over the last 3 decades that they can stimulate T cells in mice and other animal models in
Mm.
humans. Um, so there's a lot of groundwork that's been laid with mRNA technology over decades, but really in earnest over the past, well, leading up to it, of course, there was all the groundwork with thousands of people involved, but because it just came into the public consciousness a few years ago, that's why it seemed so new and so scary.
And so, over that whole development period, there are a number of significant challenges which the scientists faced in terms of making mRNA therapeutics viable, including stability. I mean, what are some of those challenges when it comes to developing and manufacturing mRNA?
Biggest challenge is making sure that the mRNA would be taken up by the body and not
Mm.
quickly degraded before it could deliver its message, which is the mRNA, which is the RNA transcript, to be read into proteins in the cell. So the mRNA molecule typically has a lifespan of very fleeting, is the answer.
Hmm.
If you took a naked mRNA molecule and just had it on the table, lifespan would be seconds, or maybe minutes to hours. Sometimes in vivo, it's considered to have, up to a 5 hour lifespan, but very short. So getting around that's a technological hurdle. And then, of course, getting it into cells before it gets consumed and degraded by the body is another hurdle that was it had to be circumvented because otherwise, you've got this potential great idea for how you can influence the internal machinery of cells, but like a speech that doesn't drive the audience to do anything, you end up kind of just talking to yourself. Well, it's kind of the same
Hmm.
thing. If you have a great technology, but it can't deliver what it's delivering to the site where it needs to deliver it, then nothing comes out of it. So
Yep, yep.
the solution to that problem came from advances in nanotechnology, actually. So the idea was these fatty droplets, lipid nanoparticles, and they wrap the mRNA like a bubble and allow it to enter the cells. And then once inside the cells, the mRNA message can be translated into proteins
Mm.
like the spike protein for SARS-CoV-2. And then the immune system would be primed to recognize foreign proteins.
Very interesting. I don't think we can really discuss this topic without addressing some of the controversy which goes around it as well. What are some of the main concerns people seem to have around mRNA technologies, and how do you respond to them as a scientist?
Yeah, it's hard. So some may ask or have seen on social media that mRNA vaccines aren't really vaccines.
Hmm.
So I guess to start with that question, is an mRNA vaccine really a vaccine? And there's a lot of misconception about that. So a vaccine is really a substance used to stimulate immunity to a particular disease or pathogen. And the idea of vaccination and vaccines came into use around 1800. And the idea was it was at the time the action or process to prevent smallpox by injecting people with cowpox virus. And the cowpox virus is variola vaccinae. And so when Edward Jenner was doing these studies for this technique that he publicized, the name vaccine essentially means pertaining to cows from the
Ah,
Latin vaccinus
oh, I didn't
or
realize that.
or vaca, which is
Hmm.
cow. Yeah.
Hmm.
So, and that's because of the first association with smallpox vaccination. So, yes, there's no reason why, because you're preventing a particular infectious disease or pathogen, that it wouldn't be called a vaccine. Um, in the case of the COVID-19 vaccine, the idea was you have an RNA which is endowed to be rapidly translated into non-functional SARS-CoV-2 spike proteins. And these trigger the immune response. And so the hope is that if that works effectively, you become immunized. So this kind of goes back to what's vaccine all about? How did it get its name? But vaccination is the term used for getting the vaccines. That's having the injection or taking an oral vaccine dose.
Mhm.
And immunization refers to the process of both getting the vaccine, but then important requirement is becoming immune to the disease following the vaccination. So
Mhm.
vaccines, there's a little time lag there. Vaccines take some time to work because your immune system has to be primed and needs time to produce an immune response to the vaccine. So the real difference between vaccination and immunization is based on the time effect necessary to provoke an appropriate immune
Right.
response. So I guess back to the controversy, the idea there's the debate was there's a lot of fake news about vaccination due to anecdote, and people are oftentimes impressed by single tales of sporadic cases of adverse events without being correctly informed about the actual risks to undergo interventions. So there were risks to any medical intervention. The CDC had reported, the US CDC reported 6 cases of anaphylaxis due to the COVID-19 mRNA vaccine, and that was back on 270,000 something doses. But undergoing surgical intervention uh, for elective surgeries has a mortality rate of it's 0.17%, and maybe up to 10 times higher during emergency surgery.
Mhm.
So, nothing in the medical and healthcare field is risk-free, but I think a lot of the issues stem from social media and reports about anecdotes. So people read and take these anecdotes and then they confabulate what would be described probably as anecdata. And the
Mhm.
problem is 1,000 vociferous anecdotes do not constitute objective data. Or as the saying goes, the plural of anecdote isn't data. So I
No.
think it all goes back to we have to have still, and we did, and we have well-run clinical trials for mRNA technologies and all medical interventions. And even then, that's potentially but one or a handful of well-run, thoughtful trials. And it's not until you have many trials that we can start amassing data and look at it meta-analytically to iron out trial-to-trial inconsistencies and biases and get to what the facts really are in there, which sometimes don't begin to appear until commercialization and released to the public, when then many millions of disparate people are exposed to the treatment. And that was what happened with Janssen's COVID-19 vaccine, if you may remember. So
Mhm.
back in 2021, April 13th, 2021, the US CDC and FDA recommended a pause in the use of their COVID-19 vaccine because they had reports of thrombosis with thrombocytopenia syndrome, which is really a very rare condition, which is characterized by low platelet counts and thrombosis, which includes unusual sites like the cerebral venous sinus after receipt of the vaccine. So that, I believe, led to, at last count that I remember, 9 deaths out of 18 million people who were vaccinated.
Mm.
So again, very serious side effect for which there was a pause in its use and then it was eventually discontinued. But that again was a handful of cases out of 18 million people who received the vaccine. Um,
Yep. Yep.
Kind of getting off the mark though. So the Janssen, just to be clear too, the Janssen COVID-19 vaccine was not an mRNA vaccine. But on the mRNA side of things, there are still data being gathered and analyzed and, new technological innovations which will help us stay at the forefront of other mRNA approaches to medicine.
Yeah, yeah. What temperature stability challenges are there in the distribution of these types of vaccines, and how is it possible to address that?
Well, mRNA typically has a shelf life of, we'll say under a year, 6 months at
Mm,
minus 80 C, minus 80 to minus 60 C. Um, Much shorter if you're talking about uh, 2 to 8 degrees C, for example. And, again, at room temperature, hours, if that. So, ultra cold chain is typically a requirement unless there's some sort of process by which the reconstitution allows the material to be stored separately from the final finished dose. But uh, yeah, it is a challenge because what we found during the pandemic was that a lot of different countries and within countries, different regions didn't have the capabilities to manage ultra cold chain supply chains. And so that posed particular challenges for how do you ship mRNA
mm,
therapies into certain regions and have them be stable for long enough for them to be able to exert their medical effect. Um, I, as far as I understand, because RNA has ribose carbohydrates, it's more unstable than DNA because it has a hydroxyl group associated with it. And so it's more susceptible to hydrolysis and destruction. And that's why its lifespan, as I said before, kind of sitting out on the countertop is incredibly short. So
mm,
future innovations here include how do you keep these products stable for longer so that you either have ultra cold chain everywhere these need to be distributed to, or you come up with some kind of technological innovations where you don't need to have ultra cold chain everywhere throughout the thread, including the last mile problem.
Interesting. So, talking about future looking, um, what excites you most, Ben, about the future of mRNA therapeutics? And are there any other diseases or conditions which you think could be tackled with this technology in future?
Yeah, I think, there are a lot of potential breakthroughs being looked at now.
Mm,
mRNA, I would say oncology represents, of course, a huge slice of the pie in the life sciences and healthcare industry, in particular, the pharmaceutical industry, medical device industry. Um, Oh, so we can't overlook that. And I think because there are so many different types of cancer, any oncologist will say cancer is not just cancer. And because they all have different flavors and frameworks, causes, molecular causes and contributions, that having mRNA technologies available to help hopefully eradicate, diseases like cancer will allow us to add to our armamentarium uh, another avenue for potential treatment.
mm,
So certainly we have lots of therapies now that over the past decade and longer, but over the past decade, enormously successful immunomodulators for cancer therapies, and newer and more advanced chemotherapeutics and targeted therapies and different antibodies, bispecific antibodies, et cetera. But being able to go after a variety of diseases, including cancer, using a different avenue will at least allow us to start to pave the way and pioneer where is this working very well? Where is it not working very well? And then in those cases, find out how it can be improved, or maybe there are some molecular mechanistic reasons why its future just won't be in those directions, but then
mm,
take the trajectories where it's working well and improve on that. So, how could you tailor it for cancer therapies, and other ways to revolutionize healthcare? It could be infectious disease treatment, which is being not only looked at now, being trialed, or for immunotherapies, for genetic disorders, for regenerative medicine. So there's a lot of prospects out there. You can look on clinicaltrials.gov, and that site doesn't have everything that exists out there, but it certainly has registered clinical trials where this technology is being used. And there's a lot of progress and prospects for the next generation. So I think the future of mRNA technology continues to be bright. And again, it's not new. You know, as much as the controversy is these aren't really vaccines, which I think we've addressed that they are, or that these were too new and they were never trialed for long enough. There have been prostate cancer, mRNA therapies developed and trialed, Ebola, among many others. So it's not like these were just invented overnight for COVID-19 vaccines.
The thing we haven't touched on, Ben, of course, is where Bill Gates sticks his 5G tracking chips when it comes to vaccines.
Yeah,
You didn't cover that in your definition.
No, I didn't. They have to get really small, and you have to make sure you're near the 5G cell tower.
Ben, thank you for so much. Really interesting to get your overview. Thank you.
Absolutely. Thank you.